Good evening, everyone. I hope you all are doing well. So, today's session will be based on case study. So, without taking much of your time, I would like to invite Mr. Rupinder sir to continue today's session. Sir, the stage is yours. Thank you.
Hello, good evening. Hello,
Very good evening, sir.
hello. I'm audible.
Yes, sir.
Okay. So, due to some workload, okay, and due to my health issue, I just लास्ट दो सेशन नहीं हो पाए वैसे टुडे दी फाइनल सेशन ओके लास्ट सेशन आज हमारा एंड दैट सेशन आई थिंक will cover the all the we can say last aspects of the DNA. Okay, DNA extraction and other things. Okay, just wait. Okay, now is it visible? Hello, am I
Yes, sir.
audible?
Yes,
Yes,
sir.
sir.
Okay, presentation is visible. Okay, okay, okay. Just wait. Yeah, so we were on the topic of the DNA extraction. Am I audible?
Yes, sir.
Yes, sir. Yes, sir.
Okay, okay. Now, but please ask, tell me what are the different methods.
Sir,
We can say
in
you are asking about the indirect
yes,
method, na?
sir.
So what are different
सर
type of indirect method you
इनडायरेक्ट
are
मेथड्स
asking?
जैसे कि लाइक मैंने कॉम किया एंड देन मेरी ही किसी सिस्टर ने सपोज उससे कॉम किया एंड देन उसके मेरे बाल जो हैं वो रूट से निकले एंड
हम्म
देन मे बी वो
हम्म
किसी क्राइम सीन पे गई और वहां पे उसके जैकेट पे या
हम्म
क्लोथ्स पे जो बाल थे वो वहां पे रह गए बट इनडायरेक्टली उस क्राइम सीन से मैं
हम्म
कनेक्ट हो गई बिकॉज मेरे हेयर्स जो थे रूट्स के साथ वो मिले हैं
Okay. So basically, not of only yours means आपकी सिस्टर या आपकी फ्रेंड भी मीन्स कॉल पे तो उसके भी हेयर वहीं पे होएंगे ना एंड वी आर नॉट गोइंग टू लाइक गोइंग थ्रू एनी इन्वेस्टिगेशन वन एविडेंस we have to investigate every aspect of the we can say part of the evidence if you are talking about the hair sample okay here then first of all hair sample हम तभी यूज करेंगे बच्चे if there is no biological sample like biological sample means body fluid evidences okay such as blood and other body fluids okay so
ओके सर
yes it can mislead the investigation. Okay, it can mislead the investigation. You are directly or indirectly can be involved in that crime. Okay, बट
ओके सर
ऐसा नहीं है कि आपको उसमें फिर फंसाया जाएगा ऐसा नहीं कि जस्ट ऑन दी वी कैन से दैट कि जस्ट कॉम के ऊपर हेयर मिलने से ही आपको एज अ कल्प्रिट या फिर अक्यूज पढ़ा जाएगा नहीं ऐसा नहीं है
ओके सर
different circumstances आपका LOA check किया जाएगा means you are present when the crime was happening okay on the time we can say time of crime wherever you okay you are in the
यस
office
सर
you are in the we can say any playing we like doing the activity which you are in with the other friends okay then आपकी स्टेटमेंट के ऊपर भी कोई इन्वेस्टिगेशन होएगा ना ऐसे ही कि जस्ट फॉरेंसिक रिपोर्ट बन गई एंड टू टाइप ऑफ डीएनए रिकवर्ड कि हाँ दो तरह के हेयर है वहां पे कि आपका भी डीएनए मिल गया और आपकी सिस्टर या आपकी फ्रेंड का डीएनए मिल गया एंड ओके फॉरेंसिक रिपोर्ट ने बोलती है देन यू विल बी कन्विक्टेड नो ऐसा नहीं होता है investigation is a very big part of the we can say any crime scene investigation okay investigation होएगा उसके बाद ही आपका ट्रायल चलता है ना कोर्ट में
यस सर
जो वो तो बहुत दूर की बात है वो तो बहुत दूर की बात है फर्स्ट ऑफ ऑल मोस्टली जो नॉर्मल अपने घर होते हैं इन द मिडिल क्लास फैमिली वी कैन से ओके सिंगल को मोस्टली है ना ऐसा तो नहीं
सॉरी
कि
सर
कोई
आपकी
ज्यादा
आवाज बीच में अटक गई थी
Aina, I'm audible.
Yes, sir.
ओके, मैं ऐसे बोल रहा था कि ऐसा नहीं है कि मीन्स मोस्टली जो हमारे घर होते हैं ना, गांव वगैरह के जो घर होते हैं, वहां पे ऐसा नहीं है कि एवरी लाइक मेंबर ऑफ द फैमिली यूजिंग अ डिफरेंट कॉम। अगर
Yes,
आप इफ
sir.
यू आर बिलोंगिंग टू एनी विलेज हाउस है ना, ऐसा तो नहीं है कि आप जस्ट उसी कॉम से ही आप मीन्स हर एक इंडिविजुअल इंडिविजुअल अपना एक कॉम वगैरह, ऐसा तो नहीं होता ना,
जी.
है ना। वो डिफरेंट कॉम कब होएगा ना, व्हेन यू आर लाइक मोर सोफिस्टिकेटेड अबाउट योर थिंग्स, दिस बिलोंग्स टू मी ओनली, my comb, my hairband, my shoe, my pant, my towel, my brush and everything and all जो भी होता है ना। बट ऐसा नहीं है, नो। अगर हेयर सैंपल है नोट ऑन द हेयर सैंपल कैन बी अ कोरोबोरेटिव एविडेंस इन एन इन्वेस्टिगेशन। ओके, इट इज द प्राइमरी एविडेंस। फर्स्ट ऑफ ऑल द
Okay, sir.
प्रेजेंस सीसीटीवी, आई विटनेस, वो भी देखे जाते हैं। फर्स्ट लास्ट लास्ट सीडीआर रिकॉर्ड दिखाता है, कॉल डिटेल रिकॉर्ड दिखाता है, द लास्ट फुटेज। Okay, what is we can say relation to the victim or we can say to the accused, क्या relation रहेगा, और what is the relation to the crime scene। Okay, जो crime scene हुआ है जहाँ पे हुआ है, then what is the relation of yours, वो भी देखा जाएगा। Okay, yes, it
Okay.
can mislead the evidence and it is we can say अगर कोई ऐसा करेगा ना, then it is called tampering of the evidences। वो इतना शातिर, इतना मीन्स sharp minded होएगा, that a person who is committing the crime, that he or she will we can say plant the another evidences to disguise him or self or herself। okay, अपने को disguise करने के लिए वो कुछ भी कर सकते हैं। okay, there are so many we can say sharp minded criminals are there in our society। clear।
Yes, sir. And sir, अगर जैसे बायोलॉजिकल फ्लूइड मिलता है, फॉर एग्जांपल सलाइवा, तो उस चीज में भी पहले बाकी चीजें पहले रखी जाएंगी एंड सलाइवा को कोरोबोरेटिव एविडेंस की तरह ट्रीट किया जाएगा।
Yes, saliva actually क्या है ना, saliva is not visible like at the that type of that extent, means the, we can say कि saliva is just a liquid, है ना, just a transparent liquid। जब हम किसी क्राइम सीन पे जाओगे ना, देयर इज वी कैन से, अभी तक तो ऐसा सिनेरियो चलता है, क्राइम सीन पे जाते जाते आपको 3 से 4 घंटे निकल जाते हैं, और उतने समय में सलाइवा मीन्स आपको आंखों से दिखाई दे जाए, इट्स नॉट पॉसिबल।
थैंक यू सर।
ओके, हाँ, मीन्स सलाइवा अगर आपको मिलता भी है देन इट इज आल्सो कोरोबोरेटिव एविडेंस, नॉट अ प्राइमरी एविडेंस। एंड सलाइवा अगेन वही थिंग सेम, कि व्हाट इज द रिलेशन with the victim or what is the relation between the crime scene and between the you or we can say your friend। ओके, हर
ओके
चीज को
सर।
देखा जाएगा उसमें। हाँ, तो प्लांट करने वाला तो कुछ भी कर देता है, प्लांट करने वाला तो, आई एम जस्ट डिस्कसिंग अ केस, सिंगल केस, एक सिंगल केस था। देयर वाज अ वी कैन से देयर वर टू पर्सन, मींस कपल था एक। ओके, एंड दैट वी कैन से द लेडी वाज ऑफ क्रिमिनल माइंड। ओके, द फीमेल पार्टनर वाज ए क्रिमिनल माइंड। उसने क्या किया कि व्हेन दे आर इन द वी कैन से इन द क्या बोलेंगे उसको, कि व्हेन दे आर इन द क्लोज रिलेशनशिप। ओके, उसने पहले ही प्लांट कर लिए एविडेंसेस with the contraceptive they used during their sexual intercourse, and they just preserve she just preserve that contraceptive। ओके, उसने अपने पास रख रखा, एंड आफ्टर द वी कैन से कमिटिंग एनी क्राइम, उसने इसको बंद कर दिया, सिम उसके वहां पे जाके गिरा दिया। ओके, देन वो प्राइमरी एविडेंस बन गया दैट द पर्सन, ओके, द semen belongs to that person is a primary suspect of that crime scene, of that crime। क्लियर।
यस सर।
सो बहुत सारे सिनेरियो होते हैं, डिपेंड्स ऑन द डिफरेंट टाइप ऑफ सिनेरियोस। ऐसा नहीं है कि हर एक, और ऐसा क्राइम सीन आपको बहुत ऐसा तो वैसे कभी मिलता है कि जैसे yesterday आई वाज ऑन द क्राइम सीन। ओके, आयुषी ने आपको बताया होगा बिकॉज क्लास इसीलिए नहीं हो पाया था।
यस
क्लियर।
सर। यस सर। थैंक यू।
एनी फर्दर क्वेरी। ओके, एनी फर्दर क्वेरी। सो आई एम ऑडिबल।
Yes, sir. Yes, sir.
ओके, ओके, The radiation visible है सभी को?
Yes, sir.
Yes, sir.
Okay. So which is the diagrammatic view can we see like that? The double stranded DNA first of all denatured at 94 degrees centigrade. Then the primers will add up and the parent chain of we can say parent strand of the DNA, and after that, after the annealing, it will lead to the extension of the DNA fragments. Okay. So this is the first cycle of the PCR. Clear? Clear?
Yes, sir.
Yes, sir.
Hmm. So now after the PCR, the next step is DNA separation. Okay, DNA extract कर लिया. After DNA extraction, DNA quantification done. After DNA quantification, PCR done. Multiple copies are found on the desired set of the DNA. Then after the amplification of the DNA, the last step of the we can say its technology is DNA separation. Okay, so the PCR product in which the STRs, means short end and feeds are formed, will produce a mixture of DNA. Now we have a mixture of large number of fragments of the DNA. So it will lead to the we can say challenge. Okay, and separation of these DNA fragments is not very easy. Okay, so for that we have different technique, that is electrophoresis. To distinguish the various molecules from one another, and a separation step is required to pull the different sized DNA. So the electrophoresis technique will separate the DNA according to their size under the influence of electricity. Okay, so electrophoresis is of different types: sludge electrophoresis, capillary electrophoresis, polyacrylamide electrophoresis. Okay, primarily we used gel electrophoresis for the separation of DNA because we don't have any technology or any advancement in the technology. Nowadays we have a capillary electrophoresis. Okay, and that part of the machine is called genetic analyzer. Okay, genetic analyzer बोलते हैं उसको, and that genetic analyzer contains the is based on the principle of capillary electrophoresis. कोई ज्यादा बड़ा पहाड़ नहीं होता है जिसको लाइक इफ यू आर टॉकिंग अबाउट दी एनी जेनेटिक एनालाइजर जो डीएनए में काम करता होगा ना, इफ एनी पर्सन वर्किंग एट डीएनए ही विल हैव एक्टिविटी ऑन इज above from sky, like उसका आसमान से ऊंचा रखा होगा. I am working in the genetic analyzer. I am working on the genetic analyzer. It's nothing. It is just a simple instrument which is based on the capillary electrophoresis. Nothing else. Okay. So PCR products are from the short and period DNA must be separated in a fashion that allow each allele to be distinguished from the other alleles. A size-based separation method is called electrophoresis. Electrophoresis is the technique in which the DNA fragments get separated according to the size under the influence of electricity. This is the definition of electrophoresis. The separation medium may be used in the form of slab gel or capillary. Okay, the word electrophoresis comes from the Greek word that is Greek electron, that is charge, and the Latin for means bearer. A charged bearer particle. Okay, the process of electrophoresis is referred to as the electrically charge carried by the molecules. In the case of DNA, the phosphate groups on the backbone of the DNA molecule readily gives up their positive charge. वो क्या करते हैं? The DNA molecule gives up their positive hydrogen ion and leaving the DNA fragment as a negatively charged in most of the buffer system. And then the negative charge will attracted by the positively anode. Okay. Under the influence of an electric field, a DNA molecule will migrate away from the negative electrode known as cathode and move toward the positive electrode, that is the anode. Clear? The higher the voltage, greater the force felt by the DNA molecule and faster the DNA moves. This last line is very much important. डायरेक्ट क्वेश्चन में वही है इस बार के फैक्ट प्लस आई थिंक फैक्ट एग्जामिनेशन में. ओके. एंड यूजीसी नेट के भी, the higher the voltage, greater the force felt by the DNA molecule, and faster the DNA moves. Okay. The movement of ion in electric field generates heat. The movement of ions क्या करेंगे बच्चे? If any electron, we can see if any charged particle is moving under the influence of electricity, it obviously will generate the heat. Okay, and that heat must be dissipated. Okay, otherwise वो क्या कर देगी? And that it must be dissipated. Otherwise, it will destroy the DNA fragments. Okay, so we need to dissipate that heat, and that heat is then absorbed by the system. Excessive heat can be cause a cell. क्या करेगी बच्चे? जो जेल होती है ना, the excessive heat can damage the gel also, and excessive heat can also destroy the DNA fragments. The excessive heat can cause the slab gel to generate extra bands, and in a very severe case, the gel can literally melt and fall apart. Performing electrophoresis in the capillary is an advantage because heat can be more easily dissipated from the capillary, which has a high surface area to volume ratio. Okay, so this is advantage of capillary electrophoresis over the gel electrophoresis. Clear? So from here, whole process is explained in this presentation. स्लैब जेल इलेक्ट्रोफोरेसिस क्या है, ठीक है, पूरा का पूरा इसमें एक्सप्लेन्ड है. ओके, एक बार इसको जस्ट गो थ्रू होएंगे, जल्दी से निकालेंगे थोड़ा सा. सो स्लैब जेल, व्हाट इज द स्लैब जेल? एक्चुअली these are nothing near the agarose gel. अगार अगार से बनी हुई एक जेल जिसको हम बोलते हैं एग्रोजेल, एंड दैट इज द स्लैब जेल. एंड व्हाट इज अगार अगार? इट इज अ सी बीड. ओके, which have pores, different pores are present in the gel. So slab gel consists of microporous matrix through which the DNA molecule pass through the electrophoresis. DNA material are mixed together and poured into the mold to define the structure of the slab gel. The sample, here we have a comb. Okay, we have a sample comb. After the preparation of the slab gel, we just insert a comb on the we can say one side of the gel, which will make the we can say small valves for the sample. Okay, a sample comb is placed on into the gel such that the teeth of the comb are embedded into the gel matrix. After the gel gets solidified, the comb is removed. Okay, and leaving behind the small wells. Okay, that will like used for the loading the DNA samples. Two type of gels are commonly used in molecular biology or forensic DNA. First is agarose gel, have very large pore size used for the separate DNA molecule, and the second is polyacrylamide gel, used to obtain the high resolution separation for a small DNA molecule, usually 500 to 1000 base pair. Okay, two type of gels we can use in the biology, molecular biology or forensic DNA laboratories. First is agarose gel, second is the polyacrylamide gel electrophoresis. So this is diagrammatic view of the electrophoresis. First, here is the anode, here is the cathode. This is the gel, and whole the container is filled with the buffer solution. Okay, to maintain the pH or to limit the heat generated during the flow of the charge, and also to make the connectivity between this whole the system. Okay, and these are the wells. Okay, and the whole the DNA strands get separated according to your size. So, what is agarose gel? Now, agarose gel basically a form of seaweed, contain pores that are of 2000 angstroms, or we can say 200 nanometer in diameter. Agarose gel easily prepared by wing. Okay, simple बच्चे process है. It's a very simple process. You are just performing this agarose gel from your I think 10th standard or 12th standard. Okay, so agarose gel easily prepared by weighing out a desired amount of the agarose powder and mix it with the electrophoresis buffer. And the mixture then quickly brought to the boil and the solution, and whereupon the agarose powder goes into the solution form. After the solution cool down, it gets settled down. Okay, and then it is poured. We can pour on anything, like जिसके ऊपर हमने जेल इलेक्ट्रोफोरेसिस करना है ना, we have to pour on that surface. Okay, on that slab it is poured into the gel box to define the gel shape and thickness. Then again, same, the comb is added to liquid agarose because before it cool to form the wells for the samples. Okay, then wells will form that can hold a sample up to 5 to 10 microliter. The number of wells formed up to 8 to 10 that can carry 8 to 10 sample at one time. Okay, this is one advantage of the electrophoresis. We can run 8 to 10 samples at one time. Electrophoresis sample is poured over the gel until it gets fully submerged. Then two buffers are used here. Two buffers mostly used in the electrophoresis. One is trace acetate buffer, trace acetate, okay, TAE, trace acetate EDTA, and second one is trace borate EDTA, TBE, okay, TAE or TBE, okay. Then samples are mixed with the loading die. बच्चे loading die से हम mix करते हैं, okay. How we visualize the samples after the separation? Okay, we need something. So that something is a die, loading die. It can be a therium bromide die, or it can fluorescent dye. Okay, a samples are then mixed with the loading dye and carefully pipetting into the each well of the submerged gel. This loading dye contain a mixture of bromophenol blue or dark blue dye. Means which there are different type of dyes are there. Okay, so bromophenol blue, a dark blue dye that help in the analysis of the C sample, sucrose to increase the sample viscosity and help in the stain the well prior to returning the voltage and initiate the electrophoresis. Okay, all are things. After the sample loaded, okay, जैसे ही हमने sample load किए, we have to cover the placed over the gel box containing the submerged gel and the electrodes on the either end of the gel plugged into the power source. Okay, then we will start the process. The anode, which is positive electrode, placed on the end of the gel farthest from the wells. Okay, because anode is the positive charge. Anode is a positive charge electrode. It will attract the negatively charged DNA molecules. Typically, 100 to 600 volt current we can say applied across the agarose gel, which are 10 to 40 centimeter in the length, creating electric field strength approximately 1 to 10 means volt per centimeter. Okay, इतना ही क्या करेगा? It will generate the electric field. Then this process will begins. The smaller DNA fragments moves more quickly because they are small size than the larger ones go through the obstacle along the gel and race course. Okay, this is the race course we can say. The smaller one runs very quickly and the larger one runs behind the that smaller one. Okay, and according to their pore size they settle down. Okay, and thus the further along when the voltage is turned off and the runner freeze where they are. Okay, जैसे ही हमने करंट को बंद किया, जहाँ पे वो थे वहीं पे रह जाएंगे. When the separation is completed, the gel is scanned and photographed to record the result for the examination comparison. Okay, this is the simple. I just put in this slide. Okay, agarose gel. But this is not the agarose gel. Sorry, for electrophoresis. Electrophoresis तो हुआ है, but which this is not for the क्या बोल रहा हूँ DNA. Okay. Actually, this gel was for the species identification. ओके, इन फॉरेंसिक बायोलॉजी डिवीजन में हम क्या करते हैं कि स्पीसीज आइडेंटिफिकेशन के लिए भी हम इलेक्ट्रोफोरेसिस करते हैं ना. आई थिंक ऑल यू हैव स्टडी अबाउट द डिफरेंट स्पीसीज आइडेंटिफिकेशन टेक्निक्स इन द फॉरेंसिक बायोलॉजी. सो दिस इज द आई थिंक वर्टिकल इलेक्ट्रोफोरेसिस फॉर द स्पीसीज आइडेंटिफिकेशन. ओके, कोई है उनमें से एक रॉकेट होती है, एक वर्टिकल एक हॉरिजॉन्टल होती है. दिस इज वन. सो इसमें क्या होती है कि आर्क बन जाती है. Okay, in this method एक arc बनेगी species identification for the species identification. Okay, one side are sample, second is this anti-zera. under the influence of electricity, they just move toward one another. Where they meet, if they are complementary to each other, they will form a arc. Okay, so this is gel, agarose gel. Then polyacrylamide gels. Again, same things are there, but the difference is polyacrylamide gel is there. Okay, it is smaller size, pore size that is 100 to 200 angstrom than the aerogel, which have 500 to 200 angstrom. The average pore size of the gel is an important factor in the determining the ability of the slab gel differentiate or to resolve the two similar sized DNA fragments. Polyacrylamide gels may be run either in the horizontal or vertical format. Okay, आपने पढ़े होगा polyacrylamide can be in both direction, horizontal or vertical. This the type of gel box to define the running format. Detection of the DNA by polyacrylamide gel may be performed with fluorescent dye or silver staining. Okay, after the running the polyacrylamide gel, we have to like if we want to detect the DNA strand, we use the fluorescent dyes or silver staining techniques. Okay, so these are the conventional methods. Gel electrophoresis, polyacrylamide. Nowadays we are using the capillary electrophoresis. Okay, it's more better than the gel electrophoresis. And now capillary electrophoresis, we can say now is in its advanced stage nowadays. Capillary electrophoresis is relatively a new addition to the electrophoresis family. The first capillary electrophoresis separation of DNA were performed in the late 1980s. Okay, nowadays this is more popular technique in the forensic science. There are number of advantages, okay, of capillary electrophoresis. First of all, the injection, separation, detection steps are fully automated. आपको मैंने already बताया बच्चे, we just we just need to put this sample in the given vials, okay, given vials, and then the whole method is fully automated. Okay, the injection, separation, reduction are fully automated. In addition, only tiny quantity of sample are consumed in the injection portion. Okay, only tiny, हमें थोड़ा सा ही sample चाहिए होगा. The separation in the capillary may be conducted in the minutes rather than hours due to the high voltage and that are permitted in the improved heat dissipation from the capillaries. Okay, the one more advantage can be done in minutes. Another advantage is the quantitative information also there. Okay, for the sample, give sample. No extra step is required for scanning or we can say detection of the at the end the we can say DNA fragments. Okay. After that, lane tracking is not necessary since the sample is contained with the capillary. We can like in gel electrophoresis, we need to trace the lane of every sample, but in this no requirement for the lane tracking, and there is no fear of cross contamination. If there is a zero, we can say chance of human error, then there is no fear of cross contamination from sample to sample. Okay, there are some disadvantages also there. Okay, अगर किसी इंसान में भी बच्चे अगर किसी में बहुत सारी we can say अच्छी आदत है ना तो जल्दी तो होगी. Same like that. If advantages are more than the one or two disadvantages are also there for capillary electrophoresis. First, the major disadvantage of capillary electrophoresis is that the time required to run the multiple sample in given time. The rate at which the sample can be processed is called throughput, since the sample are analyzed sequentially at one at a time. Single capillary. But now this limitation also overcome nowadays. पहले क्या था ना, one by one. We need to analyze one sample after the second sample, then three, then third, then four, then five. Nowadays, just put multiple sample in a tray and just we can say start the machine after hour, 1 hour, 2 hours, 3 hours, it automatically complete. Okay. Single capillary instrument are not usually capable of processing the higher number of the sample compared to the gel that captures several sample once. Okay, but this also we can see limitation. But this will, if we want to overcome this disadvantage, it will lead to the most cost price. Okay, the which price बढ़ जाएगा. Instrument required for higher startup cost. That is, मुझे आप अब तो इसका रेट नहीं पता, मीन्स करंटली जो है वो चल रहा है. ओके, जस्ट वैसी का आइडिया लिखा हुआ है फ्रॉम द रेफरेंस बुक पे से लिखा हुआ है, करंटली मोर देन $50,000. Then slabs electrophoresis. Okay, so that's why most of the laboratory don't have any genetic analyzer. Okay, काफी महंगी मशीन होने करके बच्चे most of the labs do not have genetic analyzer. Only if there is a genetic analyzer, then जो एक expert है, only one expert, which is like working from many years, okay, or we can say many months, or from many days, only that single person will work on that genetic analyzer. वो दूसरे को हाथ भी लगाने देगा, नो 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 नो, आई विल डू दिस. ओके, वो ऐसे करके बोलेगा. आप कभी जाना फॉरेंसिक लैब में डीएनए सेक्शन, जस्ट वो डेमो देंगे आपको, बट यू कांट कंट्रोल, यू कैन से यू कांट डू एनी एक्सपेरिमेंट इन द डीएनए लैब. Then what are the components of capillary electrophoresis? Simple. Primary element is the basic capillary electrophoresis include a narrow capillary, two buffer vials, two electrode connected to the high voltage power supply. The capillary electrophoresis system also contain a laser excitation source, a fluorescence detector, auto-sampler to hold the sample tubes, computer to control the sample injection and detection. Okay, it means all things are robotic. Capillary electrophoresis, capillaries are made of glass. Okay, so there is no need for the preparation of gel. The same buffer that are used in the gel electrophoresis may also be used in the capillary electrophoresis. So buffer are same in both electrophoresis. Instead of gel matrix through which the DNA molecule pass on a viscous molecule polymer solution serves as a seeming medium. Okay. Then an important difference between the capillary electrophoresis and the gel is that the electric field are on the order of 10 to 100 times stronger in with the capillary electrophoresis, which result in a much faster run than for the capillary electrophoresis. Obviously, if it is a machine, it will require more electricity and will give the more good results and stronger results. Okay, and faster results than the normal electrophoresis. So this is simple. Okay, single, एक ही कैपिलरी दिखाई है इसमें, ठीक है. दिस इज द इनलेट, कैथोड, एनोड, 5 to 20 kilovolt. This is the desktop for the reading. This is the sample tray. Here we will just put the we can say sample. This is the capillary. It will run through this capillary, and this is the detection window. Okay, laser लगी होगी इधर. Okay, so detection is the another part in the capillary electrophoresis. Detection of the sample is performed automatically by the capillary electrophoresis instrument to the measuring the time spent from the sample injection to the sample detection with a laser placed near the end of the capillary. Okay, so which is the traveling में जो time लगेगा ना from the inlet to the detection window, and that thing is observed. Laser light is shined on the capillary at the fixed position. जैसे आपको पीछे दिखाया ना, this is the fixed position. So laser will shine here. Fixed position where a window has been burned into the coating of the capillary. DNA fragments are illuminated as they pass through this window, and that illumination will be recorded by the recorder. Okay, detector होएगा. After the detector we have a recorder, and then we have a displayer. Displayer means जो हमारे पास window होएगी. Smaller molecule will arrive at the detection point first, followed by the larger molecule. Data from the capillary electrophoresis separation are plotted as a function of the relative fluorescence density observed from the fluorescence emission of the dyes passing the detector. The fluorescent emission signals from dyes attached to a DNA molecule can be used, then we use for a detector to quantify the DNA molecule passing through the detector. Okay, there another advantage is there. The fluorescent emission of the signal can also be used in the quantitation of the DNA. हम quantify भी कर सकते हैं. Okay, the fluorescent signals, with the help of these fluorescent signals, we can quantify the DNA at the end of the process. Okay, so this is the whole process. We can say diagrammatic view of the capillary electrophoresis. Here is a sample preparation. Mixture of dye labeled PCR product from the PCR. Okay, and sample injection, sample separation. Here, okay, this is the capillary according to the size. Then after that, here the argon ion laser. Okay, for the excitation. After that, fluorescence, the ABA prism spectrograph means उसको detect करेगा. After that, color separation. After that, this panel will show the peaks like this. Okay. अगर आपने कहीं प्रोफाइल देखी हो उसकी डीएनए प्रोफाइल अगर देखी हो बच्चे, इट विल लाइक अ मल्टीपल कलर की एक प्रोफाइल बनती है विद डिफरेंट टाइप ऑफ मार्कर्स विद डिफरेंट कलर्स. ओके, सो दीज आर द डिफरेंट markers which we are using in forensic lab. Some labs are using 20 markers, some labs are using 24 markers, some labs are using nowadays 28 markers. So how many these markers are there? If we are counting these, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 2, 3, 4, 5, 6, 7. Okay, so I think that 28. One push counting करना होगी. So बच्चे, the 28 markers we are using here. So first is the mallogenin for the sex determination. Then TH01. Then these are all the markers. Okay, FGA. Then there are last one Y chromosome marker होगी last one, which are only present in the male. Then TPOX. Okay, so these are the markers. So what are the different applications? Now here we end the whole topic of the DNA profiling. Any doubt?
No, sir.
Everyone any doubt? If you have any doubt, then please ask.
No, sir.
No, sir.
Okay. समझाया सभी को?
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay. So there are different applications of the DNA profiling in the forensics, such as parentage testing. There are different type of parentage testing are there. Such as paternity disputes are there, maternity disputes are there, sibling DNA testing, grandparentage testing. I'm not going to explain one by one every application. Ancestry DNA testing, genetic reconstruction DNA testing. Okay. Then in paternity, sorry, in parental testing, there is a paternity index is also there. Okay, to know the father, to know the we can say child. Okay, in which father is, we can say Alice father is there. In second case, reverse parentage में क्या होएगा? There the missing child is there. Okay, then there are number of applications in the DNA profiling are there, such as disaster victim identification, missing person identification. This is most important. Missing person में क्या होता है? If a person is missing, FIR registered होएगा. After some time, if a body is recovered under that police station area, okay, then whole the district will, we can say will active. एकदम से एक्टिव हो जाएगी and the sampling of that unknown body, if that body is unknown, okay, then उसका पोस्टमार्टम होएगा, then उसका सैंपल होएगा. After sampling, it will send to the forensic lab. And जो person missing है, जिसने बच्चे the family that reported in the police station means that person was missing from that time. Okay, the police will take the reference sample from that family anything belonging of that person, or you can say blood of their parents, of means जो भी होएगा, parents का blood होएगा, sibling blood होएगा, okay, then it will lead to the conclusion that a person, the body of जो मिली है body, okay, the unknown body is of that person or not, okay. So missing person education director reference sample हो सकते हैं. There are different type of reference sample are there. Director reference sample, family reference sample. A director reference sample means the belongings. Okay. Family reference sample, unidentified human remains. Child swapping, बच्चे बच्चा हॉस्पिटल्स में सभी देखा होगा, there न्यू बोर्न बेबीज आर देयर. ओके, कोई अदला बदली होगी, है ना, मीन्स म्यूचुअल एक्सचेंज हो गया बच्चों का. तो म्यूचुअल एक्सचेंज भी आपसे ज्यादा बदली होगी. ओके, सो सिविल इमिग्रेशन, monitoring transplants, etc. There are so many applications are there. Okay, in research field also, but genetic this DNA profiling play a very important role. Molecular biology, biotechnology, and in research field. Okay, uh, genealogy. Okay, new disease जो बच्चे आ रही है, like cancer cell identification. In that also case, in that case, DNA profiling play a very wide role, we can say important role in the whole science. Okay, not only in the forensic but also in the science. Okay, so come on the main instrument, or we can say the voting machines which we are using in the DNA labs. So this is the DNA extraction machine. कुछ नहीं करना है, just put the sample in the given, we can say cartridges or vials, and put that vials into that machine and set up like according to their software also set up karo, and after that run that machine. After some time you will go result. Okay, these are the two extraction machines. This is the Maxwell, and this is the applied biosystem की बच्चे automate express. ओके, कंपनी नेम बोला था ना मैंने आपको लिखने के लिए. सो दिस इज द कंपनी नेम, अप्लाइड बायोसिस्टम, इधर लिखा हुआ है. ओके, दिस इज द मैक्सवेल प्रोमेगा. ओके, हाँ, दिस इज द आरटीपीसीआर. ओके, दिस इज द पीसीआर. दिस इज थर्मोमिक्सचर. दिस इज जेनेटिक एनालाइजर. ओके, you will see here much genetic analyzer की तरह क्या आते हैं, some in some genetic analyzer here capillaries also, you can see also here. Okay, जहाँ पे भी होते हैं capillaries. Okay, if you see जो भी होएगा, और ठीक है. So this is the genetic analyzer. So if you want to know about the mitochondrial DNA, please check the video on the YouTube. Okay, the link is provided here. Then thank you. These are the references. And okay, any doubt?
Sir,
Yes.
sir, how SNP was do extracted, sir?
How SNPs?
Yes, sir.
Please repeat your question. How SNPs?
Sir, how SNPs do extracted, sir, from the DNA?
Same, single nucleotide proteins. You are talking about single nucleotide proteins, है ना?
Yes, sir. Yes, sir.
So single nucleotide बचे. If there are any SNPs are there, like
Yes, sir.
SNP, we can use SNP for the research work. Okay, forensic में इसका कोई काम नहीं है SNP का.
Okay, sir.
This is single nucleotide protein.
Sir,
Okay,
single
this is
nucleotide
same
polymer.
हाँ, with single nucleotide polymer protein. Okay, polymer is also a protein.
Okay, sir.
Okay. हाँ, polymer जितनी भी polymer है, there are polymers are made up of the proteins.
Okay, sir.
Okay.
Okay.
Yeah, they are simple, same method. The same method is there. DNA extraction method.
Okay, sir.
Yeah.
Thank you, sir.
What's your name?
Sir,
What's your name? Actually, I'm not able to see your name because I'm
Sir.
presentation mode.
Sir, Rishabh S.
Okay, which standard? BSc, MSc, Master,
Sir,
Bachelor's?
BSc in Forensic, second year.
First, second year. Okay, 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 okay.
Yes, sir.
Where you're studying now?
Sir, Gujarat, Parol University.
Parol University. Okay, okay.
Yes, sir. Thank you, sir.
So, anyone else? Okay, okay.
Sir,
have a question.
Anyone else? Yes.
Sir, is there a specific amount of sample required for DNA extraction, or can we get the DNA from small sample also? Like if
Yes,
we
Bache.
talk about degraded samples, can we extract DNA from degraded samples also?
Uh, yes. First answer is we need a required amount of DNA. Okay, there is a required amount. I think it's sorry, I forget that amount. Like 10 kilo Dalton. कुछ ऐसे बच्चे amount is there. There's some amount is there which we like required for the DNA analysis in the forensic. Okay, first. Second, if the sample is degraded but the DNA is intact, okay, then that sample is useful in the DNA analysis. But if the DNA is also degraded, okay, and that is not present in the that sufficient amount, okay, so that sample is not, we can say, useless. Okay.
Okay, sir. And sir,
Hmm.
which of the extraction process is mainly preferred?
Now it is a kit based method. Okay, like Poison method. What if you are doing like a manual method, then PCI method is preferably used. Okay, because due to the availability of chemicals. If you go with the Kelex, then it also like Kelex region available. If the Kelex region available, not, it depends. If you are going to the with the FT card, then you also need like washing steps, multiple washing steps. So in some case, in some labs, in not all the some labs, most of the labs, if the kit is, we can say, not available, okay, then the whole section will go with the PCI method, phenol chloroform isomyl alcohol method.
Okay, sir.
Okay.
And
Uh,
sir, DNA
yes.
profiling is always accurate.
DNA profiling is not, we can't say which DNA profiling not always accurate, because it's
Like,
a reason, because just wait, it will depend upon the expertise on the human hand, how a person working in a DNA lab is expert. Okay, if he is handling very like carelessly, है ना, the all the things very careless manner, so the things can be, we can say, क्या हो जाएंगी, गलत हो जाएंगी. Okay, go wrong, and that will lead to the contamination, and that will lead to the wrong interpretation, or we can say that will lead to the wrong result in the DNA analysis.
okay,
Okay.
sir.
Hmm. Anything else?
So I have a question. Can I ask?
Yeah, yeah, sure.
So we do have DNA techniques to identify the male DNA and the female DNA, right?
Nail DNA or
Male DNA and the female DNA. We do have DNA techniques, right, to
okay,
identify.
okay.
Yeah.
Yeah, yeah,
So,
yes, yes. Hmm.
for example, in cases where the identical twins, like one of the identical twin have committed a crime, okay, but then the suspect
Hmm.
is another's twin. So, in those cases, like, do we have any other DNA techniques where we can find out which twin exactly
No,
committed a
बच्चे,
crime?
no. For identical twins, it's a very big, we can say, limitation for the DNA, because the identical twins are the monozygotic twins. The genome remains, the whole genome of both the twins remains same. Rather, if they are a brother and sister, the whole genome will remain same. Yes, in brother and sister, if they are both twins, okay, it can, brother and sister can also be twins. Okay, so brother and sister can differentiate upon their XS and XY profile. Okay, but if the both are the brothers, both are the sisters, then DNA, like it has a big limitation for the DNA analysis.
So, apart from that, do we have any
Okay,
other techniques, sir? Like, if the
yes,
twins are like,
fingerprints.
Okay.
Fingerprints, yeah, fingerprints are unique to each and every
Oh,
individual.
there was another recent case from the Kanpur. I don't know if you know it. It was very viral on the
Hmm.
internet where two identical twins, they both were brothers. Their fingerprints as well as their retina, they both were matching for both of them. So,
Just wait,
that
just
is,
wait, just
yeah, is it
let
like
me know.
very rare cases, or do we still have any other technique to find out those kind of cases?
Please, can you repeat the case? Uh, I am noting this case.
Yes, so 1 minute. Let me just check.
I'm not aware about this case.
So it's a case from Kanpur, India, and it is
Hmm.
reported by Dr. Rahul Dev. If you want, I can send it
Reported
to
by.
in the group.
Yeah, yeah. Please send me, yeah, group, or you can send me personally. Okay,
Okay,
because
sir.
I'm not in a group.
Okay, sir. I'll send it to the Ayush sir, and then he can
Ayush.
send.
Okay.
Yeah,
Okay, okay, okay. Please send to the
sure,
Ayush.
sure.
Okay.
Yeah. Thank you, sir.
Yeah. I will read about this case. Then after if there is next any session, I will give some like after reading, I will give some suggestions, or we can say we can discuss about this case after
Sure,
that.
sir. Sure, sir. Thank you so
Okay.
much.
Yeah. Anyone else?
Yes, sir. I have a question.
Hmm.
Sir, why is quality DNA often more important than a simply having a large amount of DNA for applications such as
Hmm.
PCR or any other techniques, sir?
Actually, high quality of DNA in forensic science, है ना?
Yes, sir.
We uh, we can't expect that we will get the high quality of DNA.
Yes, sir.
We can get the large amount of DNA, but
Mm-hmm.
we can't expect that it will go for high quality DNA. High
Mm-hmm.
quality DNA means it is in the pure form.
Yes, sir.
है ना? So
Yes, sir.
for research work, we always need a high quality DNA. But in
Yes.
forensic science, we can't expect that it will be a high quality DNA because crime scene, the
Yes.
contamination is always there.
Mm-hmm.
Okay,
Yes, sir.
so in forensic science, the basic requirement is just a proper sampling from the crime scene. If there is a proper sampling from the crime scene, then it will be good results in the forensic lab.
Yes, sir.
Okay,
Yes, sir.
okay. So you ask about the two scenarios. If it is a research scenario, then the high quality of DNA is required. If it is a for the forensic scenario, then high quality DNA can't be expected, but the good we can say collection or packaging must be required.
Okay, sir.
Okay, yeah.
Okay. Thank you, sir.
Okay. So, बच्चे, before any further question, I am going to the case study. Okay. आयुष निराश हो जाता है अगर मैं केस स्टडी ना करता ना इस सेशन में. क्यों आयुष? So,
Yes, sir.
बच्चे, what were your views? क्या views है आपके? I just gave you a assignment. Do you remember?
Yes,
Yes,
sir.
sir.
Totally assignment.
Uh, whose mic is this? Buzzing sound is there. Okay. So, Bachi, do you remember this? Hello. Yeah. So, what are your, we can say, क्या था क्वेश्चन क्या था? What are the different possibilities, है ना? हाँ बच्चे, do you remember the case study which I gave you that yesterday lecture, day before yesterday? Do you remember the case study? What were the like what are the most possible क्या थे वो what are the most possible reasons that a person DNA profile, two different DNA profiles are there for a single person. What can be the possible reasons?
The first one is bone marrow transplant, and second one, from I read about it, that is chimerism, in which embryo fuse together.
Okay. Anyone else?
Yes, sir. It's a sample contamination or laboratory error, sir. In that, the blood or the tissue sample may have been contaminated or miscible during the collection or analysis. And another is a blood transfusion. In that,
Hmm.
like a blood transfusion generally does not change of the person DNA profile, sir, because a transfused
Hmm.
red blood have a known nucleus or known DNA. So only in a
Hmm.
rare cases are involving in a large number of donor,
Hmm.
white blood cell would be there, but a temporary effect, for a temporary effect only, sir.
Hmm. Why it is a temporary effect? Can you please explain why it is only temporary effect, not a permanent effect?
Sir, because it only happen in a real cases, sir. So I'm just confused in that, sir.
Actually, the lifespan of the RBC is anyone
Yes,
lifespan
sir. Yes,
of the
it's a
RBC.
120
Yeah, it's 120 hours or days.
days, sir.
Yeah. So because after 120 days,
Yes, sir.
okay,
Yes, sir.
that means after 120 days, the older red blood cells dead and new will form.
Yes,
Okay,
sir. Yes,
so
sir.
that's why blood transfusion it is a temporary,
Yes,
okay,
sir.
not permanent.
Okay, sir.
Uh, anyone else? Anyone else? I'm just taking the results from the students. Please, one by one, answer me. हेलो दो बच्चे नहीं आंसर देता मैनू सॉरी बच्चे पंजाबी आ गई दो बच्चे ने आंसर दिया है मुझे हेलो एम आई ऑडिबल
Yes, sir.
या प्लीज अनदर
Priyanshu.
आई थिंक यू डिडंट कंप्लीट योर असाइनमेंट no this is for your studies you are bachelor or master students take serious all assignments please anyone else
Priyanshu, hello, Priyanshu.
Yes, sir.
Uh, okay, so you can give answer. Okay.
yes Priyanshu
Sir, can I
okay
say?
yeah sure
Yes, sir, there is also two more. It's a massive blood transfusion, sir. In
hmm.
that, sir, basically in rare situation involving in a transfusion, like many donor white blood cells or a temporary mixed DNA profile may be detected also, sir. And
Hmm.
so the second one is a cancer affecting blood cells. Sir, in that a certain blood
Hmm.
cancers such as a leukemia can cause a genetic change in blood cells also, sir.
Hmm. Yeah sure very good very good
Thank you,
And
Bachchi
sir.
sir,
very good
and others
hmm.
are cell somatic mutation, in which like after the
Hmm.
birth, cells change, and one is mosaicism, in which after fertilization, the DNA changes.
Yeah okay very good so these are the answers very good so Bachchi the case study it was a real case study okay and it was based on the chimerism okay chimerism means recombinant DNA technology. Okay, whenever we are talking about the research biology or molecular biology or biotechnology, so we always use a term that is chimerism or chimeric DNA. Okay, and that chimerism or we can say chimeric DNA is nothing but is a recombinant DNA. Okay, this is the one condition in which there are two type of DNA. We can say one from the parent, another from the donor. Okay, can lead to the two type of DNA profiles. So based on this, like above discussion, here a case of DNA chimerism, a wrong brother case, also called a wrong brother case. The DNA from the sexual assault investigation initially matched with the incarcerated man because he had previously received a bone marrow transplant from his brother. Okay, there was a case of two brothers whose means some year ago they gone through the bone marrow transplantation. Okay, the actual source of DNA was later determined the doctor, so donor brother. Okay, so the chimerism case where DNA matched to be a wrong brother. The forensic DNA investigation for a bone marrow transplantation. Okay, this is a case of bone marrow transplantation. First of all, the DNA profiling considered one of the most reliable forensic identification tool. Okay, as you know, कि DNA profiling को हम क्या मानते हैं? It's the most reliable forensic tool. But in rare circumstances, biological events can complicate DNA interpretation. And that biological events, as you already discussed, or we can say you can already answer to me, such as mutations, such as somatic cell regeneration, such as cancer cells like leukemia cells. Okay, and there are another possibility bone marrow transplantation. There is one more condition that is whole blood transfusion. Okay, there are some circumstances in which biological events can complicate DNA interpretations. So, how we can rely on the DNA? If there are exceptions are there, then how we can rely on the DNA? So this will raise a serious question on the DNA profile. This case demonstrated how chimerism led the investigation to initially identify the wrong suspect. Okay, this will like miss. I think yesterday or today, I don't know. In the beginning of the session, I think one should ask me a question about the क्या था वो mislead the investigation वाला question पूछा था ना कि that if indirect transfer of the sample are there. So this is the best example in which the investigation we can say it will lead to mislead the investigation. Okay. So the case background was like that. A sexual assault case occurred where the biological evidence was recovered from the crime scene. The semen samples was collected from the victim and then submitted to the lab for the DNA analysis. And a DNA profile was generated and searched against the criminal database. First of all, the DNA profile matched with the previously convicted offender. Okay, so it clarified that here, according to this profile, it clarifies that okay, we go to the offender, but investigator considered the match highly significant. However, the suspect was incarcerated at a time of offense. Then the suspect could not have committed the crime due to his imprisonment, but that person was in prison. Okay, so investigation questioned the validity of DNA match. Then, then in this case, another attempt was made for the DNA profile. Further investigation became necessary now. Then re-examination of the case, or we can say reopening of the case occurred. The DNA profile from the crime scene was reanalyzed. The laboratory procedures were reviewed for possible errors. No contamination, no analytical mistakes, no other things, no human errors were found. Okay. Then after going to the medical history of the person that was in a prison, the investigation examined the suspect's medical report. They discovered that he had undergone bone marrow transplant years ago. Okay, and the donor was his biological brother. So now it will rise, we can say, big issue in that case. In that case, okay, that what is a bone marrow transplant? So bone marrow transplant contains stem cells that produce the blood cells, because bone marrow produces the blood cells. Okay, सभी को पता होगा ना कि bone marrow produce करते हैं red blood cells. So after
Yes, sir.
transplantation, the recipient may develop blood cells containing the donor cell. Means now the recipient have two type of DNA. Now two type of DNA cells are there in we can say in his body, or if it is a female, in her body. Okay, one from the donor bone marrow, and that bone marrow will produce the red blood cell. And second, DNA cells like hair cells, we can say saliva cells, other cells. Okay, now that person two type of cells are there, and that two type cell contains two type of DNA, one from the donor and other either parent DNA, and this can affect the forensic DNA interpretation. So chimerism, which is a, we can say, method or recombination method. Okay, recombinant DNA technology को ही बोलते हैं chimerism. So chimerism occurs when an individual possesses two genetically distinct cell population. The recipient's blood may contain the donor's DNA. The different tissue may therefore produce different DNA profile. Yes, that's why I asked the question that what can be possible reasons behind this. Okay, so different tissue may therefore produce different type of DNA profiles. It is not due to the contamination. It is not due to the any false or human error or false interpretation. It is due to the two type of DNA present in the recipient body. So, as per the forensic implications, the blood cells from the affected donor genetic profile. The DNA database therefore contain donor DNA instead of sorry, donor DNA instead of recipient DNA. This creates a challenge in the forensic investigator. Okay, so after that, after clarifying the doubts, okay, they identify the real suspect. Investigators expanded their inquiry to the family members. After inquiring their family members, the donor brother became a person of interest, and the additional evidence linked to him to the crime scene. Okay, then after confirming whole the scenario, okay, the DNA sample from multiple tissue were collected, such as blood, buccal swab, other biological samples were compared, and findings confirm the presence of chimerism. Okay, then accordingly, so scientific findings, so scientific reports to money. Okay, in which it included that the recipient blood contained donor-derived DNA. Okay, the DNA profile from the crime scene originated from the donor blood. I समझे be the DNA profile from the crime scene, because at the crime scene they obtained the blood. Okay, they collected the blood from the crime scene, and that blood belongs to the donor because due to the bone marrow transplant. So the original database match was therefore misleading the investigation. Was misleading the investigation. So what were the challenges during that that scenario? First of all, interpretation of mixed or unexpected DNA profile. But challenges ऐसे नहीं आते. Challenges, that type of scenarios give rise to the authenticity of the DNA report, first of all. Second, the expertise of the forensic scientist. Okay, तो उसकी expertise के ऊपर भी question mark होता है. Whenever they are summoned to the court, defense will ask about the expertise of that person. What is the qualification of that person? What is the expertise of that person? The defense will directly says to the judge that he is he is not an expert. Okay, so these are the basic challenges. Interpretation of mixed and unexpected DNA profile, recognition of medical conditions affecting DNA result. Third, importance of integrating medical history into the forensic. So this is the basic information. We can say this must be included to in the SOP, that in the investigation the medical history of every individual of means जो पर्सन वो है अक्यूज्ड है, द फैमिली ऑफ दैट अक्यूज्ड पर्सन मस्ट बी नोटेड, मेडिकल हिस्ट्री। ओके, देन कौन क्या सीखा हमने इससे? व्हाट वी हैव लर्नड, DNA evidence must always be interpreted in context. Second, investigators should consider their biological phenomena, and medical history can be critical in resolving the forensic mysteries. So, what is the impact on forensic science? It will increase the awareness of the chimerism among the forensic professionals. Development of guidelines in DNA interpretation, improved protocol for complex identification cases. Because, bache, when I was working in the FSL, okay, I face same type of scenario, means same type of question from an officer that was working in a DNA division. He asked me, रुपिंदर, एक केस है यार, उसमें दो तरह का डीएनए प्रोफाइल आ रही है, एंड आई वाज समन टू द कोर्ट, what are the possible reasons for that? Then I discussed by that officer. Okay, so these are the different cases you should know about the if you are a forensic student. Okay, so improved protocols for the complex identification cases. Conclusion: DNA evidence is powerful but not infallible. Okay, chimerism can lead to unexpected forensic outcomes. Careful scientific investigation ultimately identified the correct suspect. So, this careful scientific investigation ultimately identified the correct suspect. This line is the answer to that question which was just asked just right now. किसने पूछा था कि कौन सा that question कि good quality of DNA at a large amount of DNA, है ना? So this is the answer as per the forensic point of view. Careful scientific investigation ultimately identified the correct suspect. So these are the references. Okay, clear? So bache, this was all about the, we can say, your sessions on the DNA.
So your mic is off.
Yeah, sorry. I was asking you about that to conclude the session. Please conclude the session now.
So we conclude this session. So thank you so much, sir, for such an informative and insightful session. We truly appreciate your valuable time and guidance. Thank you all the participants for joining today's session.